So the Clinical Research Department conducts research that focuses on projects that have immediate translational importance. We've been conducting clinical trials over the last 30 years, and really we conduct everything from early phase 1 studies right through to multi-center phase 4 studies. And our areas of research are quite broad. We look at disciplines such as critical care, also malnutrition, particularly looking at mortality risk assessments, sickle cell disease, HIV and HIV-related research, as well as the neurosciences. We have other areas of growing research that we're also working on, particularly around newborn care, antimicrobial resistant, snake bite research, and also just trying to develop platforms for having pragmatic trials and also adaptive trials, so that trials can be more true to life and show effectiveness in a real life setting, but also be efficient in terms of cost and time. And we do this by having a clinical surveillance platform that's been in place since 1998, looking at reasons for severe bacterial infections in critically ill children. We also have the DSS, where we're able to locate participants in research studies within our community and able to very closely monitor and follow them up in the context of early phase trials particularly.
Our research group has been conducting research in critically ill children with our mind very much on to actually providing evidence-based guideline. While we conduct this research, we are always very mindful as to where the patients are being enrolled, but also the context in which any future guideline may be implemented, such as this emergency area within a paediatric ward in eastern Uganda with relatively limited resources. So what are the treatments we've actually looked at during this time? We led the fluid resuscitation trial that came up with definitive evidence for, in 2011 called FEAST. We were a major part of running the Aquamat trial that found that artesunate is better than the time-honoured quinine, but also more recently provided transfusion, definitive results for transfusion guidelines.
CHAIN is a network of 9 sites in Africa and Asia, and is led from the KEMRI-Wellcome Trust Research Program. CHAIN started by conducting a cohort study of sick children admitted to hospitals and followed for 6 months after discharge to identify intervention opportunities. We have found that half of deaths occur after discharge and that many children have low risk of death. The results mean that a child-centered and risk-stratified approach is needed rather than syndrome-based care only. CHAIN has also been behind identification of a risk stratification working group at WHO. CHAIN has evolved into a clinical trial platform, conducting biological and psychosocial interventions.
Antimicrobial resistance poses a serious threat to neonatal survival in sub-Saharan Africa, where neonates account for more than 50% of pediatric admissions and deaths. High-resistant rates, the WHO recommended empiric antibiotics have been reported in this region, and clinicians and policymakers are often not sure of the best antibiotic combinations to use in neonatal sepsis. The WHO is repurposing potentially legacy antibiotics such as fosfomycin for use in neonatal sepsis, given the limited availability of effective antibiotics in the pipeline. We have conducted several studies to better understand the etiology of neonatal sepsis, the burden of antimicrobial resistance, and to also investigate potential antibiotics for use in neonatal sepsis. This includes the NEOBAC study, which is a blood culture surveillance study that is conducted in 3 level 4 county hospitals in Kenya, and the NEOPHOSPHO clinical trial, which showed that fosfomycin was well tolerated among neonatal sepsis and provided dosing recommendations for use based on PK modeling done. The NEOPHOSPHO trial highlighted the challenges faced in conducting PK trials in the very sick and the very small neonates, given that there are limited centers with capacity for early phase neonatal PK trials in our setting. Following the NEOPHOSPHO trial, we participated in the NEOOB study, which is a large multi-site cohort study that generated robust evidence on the clinical features, pathogens, and outcomes of sepsis among neonates hospitalized in 19 centers in 11 countries. Our next steps involve the conduct of a large multi-country clinical trial, that is the NEOSEP study, And this study is going to investigate both new and currently used antibiotic combinations and provide ranking of clinically relevant antibiotic regimens for neonatal sepsis based on the efficacy and safety. Inclusion criteria in this trial is based on a novel neonatal sepsis severity score, which was derived from clinical features that are associated with high risk of mortality in the NEOOB study. Using routine admission data at the Kilifi County Hospital, we have been able to validate the performance of the NEOOB's mortality risk score and also test clinical criteria predicting meningitis among neonates and young children. As seen, the combination of our long-term surveillance of invasive bacterial infection and antimicrobial resistant lab work, which includes environmental swabbing, and clinical research conducted at our various sites, all have great potential in informing infection prevention and control strategies and antibiotic treatment guidelines in the future.
So the work that we are focusing on in regards to vaccine development spans from preclinical development all the way to testing the efficacy of vaccines. In the preclinical development space, we're looking at antigen discovery. So we have identified a couple of malaria transmission blocking antigens that we have tested in animal models and looked at efficacy and immunogenicity, and the potential there is to then escalate these to potential further vaccine development. In regards to testing efficacy, particularly the work that I am doing is establishment of the human infection studies. And for this platform, we have the malaria challenge studies, as well as we're soon to onboard a Shigella challenge study. Within the human infection studies platform, we are looking at antigen identification of correlates of infection, as well as correlates of protection in the context of testing vaccine efficacy.
We work on diseases that are a priority in Africa, and these are diseases that affect all ranges of populations, adults, children, pregnant women, and so forth. And our focus is to develop solutions that can provide protection against the common diseases. So, for instance, we previously contributed to the development of the Ebola vaccine that's now licensed, and that was a first-in-human study conducted at the KEMRI-Wellcome program, which speaks to our capacity to conduct the highest level of clinical trials, starting from very early to the late stages, which is now around vaccines that we are working on for yellow fever and fractional dosing of pneumococcal vaccines. So some of the vaccines we work on include one that I lead on, which is Rift Valley fever vaccine. This is a novel approach where we are using the same product for humans and animals, and the vaccine has already undergone advanced clinical trials in livestock and is just completed enrollment in human phase 1 studies in Oxford, and the safety looks good so far.
Neuroscience is a very multidisciplinary team, and neuro research is sold as the program, because we started in the 90s when the program started, focusing mostly on the bedside neurological manifestation of illnesses presenting to the ward. I think the work that we are known mostly of is conducting very large studies of a prevalence of epilepsy and other neurological disorders, but recently we've been able to conduct prevalence of mental health problems, especially in children, and also prevalence of neurodevelopmental disorders. We are conducting a mental health awareness program supported by the Wellcome Trust called the Diffusimo. So it has had a lot of impact, reaching over 2,000 people in the community and around 1,900 students engaged through the program. So we are quite having a lot of impact, and through that we've actually realized the need to conduct mental health interventions, specifically the MH gap, so in which we are training the healthcare providers so that they can be able to identify and manage patients with mental and neurological disorders.
Sickle cell anemia results in about 300,000 deaths annually, with the majority of these deaths happening in sub-Saharan Africa. And for Kilifi, we have about 0.8% of the population being born with this genetic condition. And our research interest as a group is to define the clinical epidemiology of the disease as well as the management that these patients receive. So part of the work that's currently ongoing is that we provide diagnosis for sickle cell anemia, both for different research studies that are ongoing, but also for clinical admissions at the wards. We also run a sickle cell clinic at Kilifi County Hospital for the management of the condition.